So I just wanted to again say thank you so much to Defeat Duchenne Canada, especially to the Research Funding Advisory Committee and the Duchenne Family Advisory Committee for their continual support of our research program. And it's really an honor to be presenting our project to you tonight. So in my lab at McGill University, my lab is really focused on studying the biology of these muscle stem cells, which you've heard about tonight. They're also called satellite cells. So these are highly specialized cells residing in the muscle tissue. And that's this cell that's shown here on the right-hand side that were first discovered and described in 1961 by Alexander Morrow. And you can see in this schematic that they are really a rare population of cells that are sitting on top of muscle fibers. And these cells are critically responsible for sustaining the regenerative capacity of our muscles throughout life. And so in the lab, we're interested in understanding how these cells are maintained both in the context of health as well as in disease. And so in the lab, we use different approaches to study these muscle stem cells, and I will show you some of our data for each of these approaches today. So on the left is what we call an in vitro differentiation assay, where we look at the fusion of stem cell-derived muscle progenitor cells into these multinucleated structures called myotubes. So these are the mature muscle cells that are in our tissue. And in the image on the top, we're looking at muscle stem cells that are cultured on top of an isolated myofiber from a hind limb muscle from mice. And then on the far right, we are looking at a cross-section of a regenerating muscle, and we can stain for different muscle stem cell and progenitor populations and get a sense of how well the muscle is regenerating by measuring these different parameters, such as muscle fiber size. And so a main research focus in the lab is how are muscle stem cells affected in Duchenne muscular dystrophy. And there are, in the literature, increasing evidence showing that these stem cells, which are lacking dystrophin expression in this disease, how they are impacted and how they don't behave the same way as normal and healthy muscle stem cells do. And so in the lab, this is how we think about DMD. And really, in essence, we think of it as this two-pronged disease where the absence of dystrophin in the muscle tissue leads to muscle fiber instability, resulting in myofiber damage, but that the lack of dystrophin in the muscle stem cell also is contributing to this disease. And so we view DMD as both a disease of the mature muscle as well as a stem cell disease. And so really the rationale for our project came from previous work that was done in the field of muscle wasting in cancer-induced cachexia, which is a severe muscle wasting disorder, where treatment of both cells and mouse models of cachexia with a drug that induced formation of these cellular entities called stress granules resulted in improved muscle differentiation and function. And so we asked a very simple question: would this work in DMD cell and animal models as well? And so, as I mentioned, stress granules are these types of cellular structures that are known as MRNP granules. And what we know about MRNP granules is that they actually play an important role in stem cell differentiation. And so our hypothesis is that if we can drive the formation of these stress granules, we would be pushing the stem cells towards differentiation. And so that's exactly what we saw. So here's an example of that in vitro differentiation assay, where we treated DMD myoblast progenitor cells with compounds that induce stress granules and performed this differentiation assay. And what we see is improved formation of these myotubes upon treatment with these compounds. And then, of course, we also looked at the muscle stem cells. So these are cultured on top of single myofibers. These are fibers isolated from DMD mice. And here, what we're looking for is the ability of stem cells to turn on muscle differentiation markers. So that's myogenin here, shown in green. And what we found was that treatment with these compounds resulted in an increase in the number of these green cells. So that means that we were seeing an increased differentiation in DMD muscle stem cells with this compound. And so finally, we have some really exciting pilot data in DMD mouse models. So here we're using the commonly used MDX mouse model, and we're trying to find the right dosage for the compound. And what we found was that with the compound, we're seeing improved regeneration, and we were able to measure this using different types of measures, including staining, as well as measuring fiber size. And so our current research project that was just funded will continue these studies. So we're looking at examining the exact expression of these MRNP granules and these genes that regulate stress granule formation in single cells from DMD mice, muscle stem cells isolated from DMD mice. And really, we are interested in understanding what is the molecular mechanism behind this compound. How is inducing stress granules able to enhance myogenic differentiation? And most importantly, we are going to continue our studies in our DMD mouse models, but also using patient-derived cells to validate our findings. I want to acknowledge my research team at McGill University. So these are really the people that are driving the work. These are the members of my lab. I also want to thank my partners for partnering with me in this work. So Stem Cell Network in Canada and the McGill Regenerative Medicine Network that are partnering with me in this research. So finally, I want to thank also our collaborators that have supported this project. These are the different funding agencies that support different aspects of research in my lab. And again, these are the names of the research team members of my lab. So thank you again very much for giving us this opportunity.